Alright, hi guys. I'm back with another video because I've been neglecting these. Alright, so today we'll be covering photosynthesis, and this is like a really hard test for me personally. And let's just get started. So first, the scientists that you need to know are, I don't know how to pronounce his name, Jan van Helmont. And then you can just quiz yourself right here. And the correct answer for who Jan van Helmont is he was the one that noticed that the mass of the tree increased due to water, and he was one of the first to use quantitative measurements. So our next scientist is Joseph Priestley, and he was the one that noticed plants produce something to keep this candle going, and we now know that the thing the plant produced is oxygen. Our next scientist is Jan Ingenhousz, and basically what he noticed was that oxygen was only given off by leaves during the day and not during the night. And at night, carbon dioxide was given up. So basically, he was the one that noticed that oxygen is given off during the day and carbon dioxide is given off during the night. And then our last guy is the most important guy, and he's Melvin Calvin. The Calvin cycle is actually named after him. And basically, he tracked the pathway of carbons as it was forming glucose, and he discovered the light independent reaction, and that's why the Calvin cycle is named after him. Okay, so let's start covering the LDR and the LIR. So the LDR is also called the light dependent reaction because it depends on the light from the sun. And technically, the light independent reaction also depends on light from the sun, but just not directly. So this one, we have the sunlight, and then the photons, which are like particles from the sun, directly hit the chloroplast. And once it hits, so we have these electrons, and then once the photons hit the chloroplast, these electrons get very excited, and they aren't actually going up in this image. Like if you see an arrow pointing up, it just means the electrons have gained energy. So the light from the sun gives energy to the electrons, and now that it has energy, it starts moving through these carriers. These are called electron carriers. And as it moves through the electron carrier, it loses energy. And part of that energy is used to power this hydrogen pump, which we'll get into later. But the electrons lose energy as it goes to the electron carrier until it arrives in photosystem 1. So this is it started in photosystem 2, and then it goes to photosystem 1. And then the photons hit photosystem 1's chloroplast again, making the electrons excited and gain energy again. So basically the photons give the electrons energy, and then now that it has energy, it starts moving through these electron carriers again before finally being joined with 2 NADP plus and 2 hydrogen ions. So that H plus means it's a hydrogen that's positively charged, which is a proton. And then combining all of these, we get 2 NADPH. NADPH is basically, it's kind of like ATP because it's a energy carrier, and this will be needed in the light independent reaction. All right, so now let's go through the small details of this. So now we know how the electrons move, but since you notice, since the electrons are moving this way and it's getting turned into NADPH, but how do we get new electrons to repeat this cycle? So we get new electrons, we replace the ones that we lost over here through a process called photolysis. And photolysis, basically, it breaks water. So let's say we have 2 H2Os, 2 waters. Photolysis will break it into oxygen and 4 hydrogen ions and 4 electrons. So basically, the two O's from 2H2O become O2, which is oxygen. And then we have 4 hydrogens, right? So we take the protons of the hydrogen and we turn it into hydrogen ions. And then we take the electrons and we just put it into this electron carrier. Okay, and then now that we have these extra ions, like, also remember how, like, as the electrons move through these electron carriers, it loses energy. When it loses energy, it's used to power the hydrogen pump. So this hydrogen pump will pump hydrogen into the thylakoid space. So this over here is called the stroma, and this over here is called the thylakoid space, also known as the lumen. So, like, there's hydrogen ions going this way, and there's hydrogen ions going this way. So there's more hydrogen ions in the thylakoid space compared to the stroma, which means we have a concentration gradient. And that also means these hydrogen ions, since they're going to the place where there's already a lot of hydrogen ions, this is going against the concentration gradient, which means this is an active transport. So this requires energy. And we get the energy from the electrons over here, like as it's moving through the electron carriers. You see, it's losing energy. That energy goes here to power this hydrogen pump, which moves the hydrogen ions actively to the lumen. Okay, and then now, since there's a concentration gradient, the hydrogen ions over here, this thing is called ATP synthase. And since, like, there's a concentration gradient, remember? So the hydrogen ions would want to go where there's less hydrogen ions. And since all the hydrogen ions are, like, over here, it'll want to move outside. So as it moves outside, it'll start spinning this protein, which is ATP synthase. And when this thing spins, ADP plus P will bind and turn into ATP. And that is the light-dependent reaction. So to summarize, light goes in, it gives energy to electrons, and these electrons travel across before finally combining with 2 NADP plus and 2 hydrogen ions to make NADPH. And then also, hydrogen ions are moving both actively, which is against the concentration gradient, and passively, which is with the concentration gradient. And then because the hydrogen ions are moving through the ATP synthase, it'll also power this, like, bond, and it'll bond the ADP plus P into ATP. And then also, through photolysis, we give the electrons that we lost back. So basically, it replaces our electrons, and it also releases oxygen. So oxygen in here isn't really a product, it's like a waste product. So it's not like LDR's function isn't to produce oxygen, but it creates oxygen as like a waste product, which means our products are oxygen, ATP, and NADPH. So we get NADPH from the electrons moving this way. We get ATP through the hydrogen ions moving passively through ATP synthase. And we get oxygen as a waste product through photolysis. Okay, now let's move on to light-independent reaction. So light-independent reaction is connected with light-dependent reaction. All right, so from the atmosphere, you know how plants need carbon dioxide? Well, this is where the carbon dioxide comes to play. So from the atmosphere, plants, like, absorb the carbon dioxide, or not absorb, but, like, they take in the carbon dioxide. And then over here, this is a 5, there's 1, 2, 3, 4, 5. There's 5 carbons, and then there's 6 of these 5 carbon structures. So say there's, like, one molecule that's made of 5 carbons. So there's 6 of those 5 molecules. There's 6 of those 5 carbon molecules. And then you combine those 6 5 carbon molecules with 6 carbon dioxide. Those 5 carbon things are called RUBP, by the way. So we combine 6 RUBP with the 6 carbon dioxide from the atmosphere, and we do that using an enzyme called Rubisco. So basically, like, Rubisco is, like, just an enzyme, and it has its drawbacks, which we'll talk about later. And then once we combine RUBP and CO2 using Rubisco, we get this. So, like, we take the carbon, so there's 5 carbons over here from RUBP, and there's 1 carbon over here from carbon dioxide. We take the carbon, we add it, and then it'll become 6 carbons, right? But then this 6 carbon structure is very unstable. So it'll break into half, which leaves 2 3 carbon structures. So this is a 3 carbon and this is a 3 carbon. And each of these 3 carbons are called pigas. So over here, there's 12 pigas. And because like 6 times 6 equals 36, and then 12 times 3 is also equal to 36. So we're just rearranging the carbons. Okay, and then now that we have 12 pigas, so remember from the LDR how we got ATP and also NADPH? Well, now we use the ATP and NADPH into the light independent reaction. So basically, ATP will break, forming ADP plus P. And remember, when ATP like breaks the bond between these two, it'll release energy. So this gives energy to that. And then also from NADPH, the hydrogens will break off and will get added into the 12 pigas. So basically, these two both like break, and then we get their stuff added to piga. And then once we add the energy from ATP and the hydrogen from NADPH, we get pgal. It's still 3 carbons, but it's just with all that extra stuff added. And then now that we have 12 pgals, we can take 2 of the pgals out, and then 2 pgals form glucose, and that is the product we want. That's how plants make their food. And then you know how we have 10 pgals left because we took 2 out? Well, these 10 pgals, like, once again, we have ATP over here, and when ATP breaks, it releases energy. So the energy release powers this reaction where it'll form like new bonds. So like there's 10 3 carbons. It'll form into 6 5 carbons. So these are still the same. It's just you rearrange the carbons, because 10 3 carbons is 30 carbons and 6 5 carbons is also 30 carbons, which means these two are still equal. We just rearrange the carbons a bit. And so basically what we get from this entire cycle is glucose, ADP plus P, and NADPH. We get glucose from the pgals. We get ADP plus P and NADP plus from over here, like when they're trying to add stuff into piga to form pgal. Okay, and then all this stuff, these two will go back to the light dependent reaction over here and get reused. So basically, to sum things up, from the light independent reaction, we get NADP plus and ADP plus P. Like, we produce these things, and then these things will be used in the light dependent reaction in order to form ATP and NADPH. So it's this ongoing cycle. Okay, now we'll talk about some problems with Rubisco. So there is a problem with Rubisco which causes some plants to like act differently. So basically, Rubisco sometimes, instead of taking carbon dioxide, it takes oxygen. But since oxygen is a waste product for plants, if it has too many oxygens, it's harmful for the plants. So different plants deal with this like problem with Rubisco differently. And this problem with Rubisco is called photorespiration. I think it's called photorespiration. Yeah. And then, so basically, there's our normal type of plants are called C3 plants. And C3 plants, basically, they just fix carbon in the Calvin cycle. So that means they add the carbon dioxide inside the Calvin cycle without adding anything new. But then we have the plants that deal with a lot of photorespiration, like the problem with Rubisco, and they have to like find a new way to solve that problem. So C4 plants deal with photorespiration by fixing carbon dioxide into these 4 carbon compounds. So that means they'll like take in the carbon dioxide in a separate compound and then move it into the Calvin cycle. And then also the stomats are partially closed during a hot day, so water won't be able to, like, you won't lose as much water. And then since there's less photorespiration, it'll grow faster. And then our other type of plant that deals with photorespiration is called a cam plant. So think of like cactus and stuff. It basically closes the stomats during the day and open it during the night. So like during the day, it'll take in the carbon dioxide, like it'll keep the carbon dioxide and organic compounds, and then it'll release it during the day. So basically what I mean is like at night, it'll take in carbon dioxide and then it'll store it in these organic compounds and then they'll release it in the day. And I think that's all you have to know for this lesson. If you guys have any questions, just like text me. All right, bye.